Folks, I'm the last one before cocktails, so we'll keep this short and sweet and also hopefully informative. This is not going to be a technical talk. I think we've heard a lot of technical talks today, amazing machine learning models, amazing data processing methodologies, et cetera. What I intended to do in the next 15 to 20 minutes, and hopefully we leave a couple of minutes for questions, is to really gear the discussion towards powerful use cases, right? So we've heard a lot of machine learning models today, but I think it behooves us as a machine learning and artificial intelligence community to really connect with our stakeholders, the business stakeholders, the clinical stakeholders, the medical stakeholders, so that we democratize access to these AI models and really enable decision making using AI and ML. I think that's what all of us want. How many of you have read this book? Can I see a show of hands? Deep Medicine? Yes, I see one. I see a couple of hands. That's awesome. If you haven't, definitely recommend reading this, Eric Topol's book. He, in the book, kind of highlights a few sort of very important things that AI is intended to address, and I think we're starting to see that become a reality. So AI will reduce healthcare costs. So one classic example is, can we actually predict mental health disorders? We heard the neuroscience talk, and can we predict them early on, right? So that we catch patients early, and I think this is applicable not just in the mental health space, even in oncology, which is my primary area of research. AI shows most promise in for clinicians with patterns. We see this especially where images are used, right? So radiological images, pathological images, and that was the first area in which artificial intelligence was really deployed in a clinical setting. So when there are patterns, I think it's easy to kind of train the machines, and we also collect lots and lots of data, and imaging is a classic example of that, and we see more and more of it. So the question in front of us is, what else can be like that, where we could kind of learn from patterns? And genomic data is extremely complex, but I think we're slowly getting there with the transcriptomics, the genomics, the methylation data, et cetera, being collected in large quantities. We're starting to recognize those patterns. So I think clinical domains with patterns really lend themselves very well for machine learning modeling. AI could be a game changer in mental health. We just said that. And we heard the amazing talk by Parth Patel 2 talks ago. AI can give back the gift of time to clinicians. I'll actually show you an example with pancreatic cancers where the oncologists were able to save time using a machine learning model to identify clinical trials for their patients. And this reduced time by 30 to 40 minutes of searching on the internet for them to identify, and the 30 to 40 minutes really amplifies. This is just for one patient, and if they saw 200 to 300 patients, and this is just one clinician we're talking about here. But there are lots of other sort of liabilities that one has to balance as we begin to think about how do we deploy this in life sciences and healthcare. Obviously, privacy and security issues. There are lots of talks in the other hallway about how we can fundamentally address some of these data breaches and ensure that there is trust fabric that's built around all of the data sets that are accessed. And it's particularly important in healthcare, right, because there is patient privacy involved, and we want to make sure that the data are fully de-identified. If I have 75 polymorphisms from you, right? This is just 75 polymorphisms. We've got 3 billion bases of DNA in our cells, right? If I have 75 polymorphisms, we can uniquely identify an individual, right? So all you need is a reference database with SNPs, and you draw blood and you do the genotyping, and you only need 75 polymorphisms. So I think the level at which the privacy really hits home is much higher in healthcare. Lack of explainability. So one of the reasons why deep learning is, somewhat, I would say not less popular, but less utilized, I guess 2 reasons. One is lack of data. So even though we talk about these large compendium of data sets, the Cancer Genome Atlas, the UK Biobank, et cetera, once you whittle it down to your use case, right? So if your use case is pancreatic adenocarcinomas, and you're specifically looking for people who will respond to a certain class of therapy, you end up with a few hundred patients or even less than that. So that's one of the reasons why deep learning has not been very widely applicable in the healthcare space, because once you kind of filter it down, the data sets end up being small. The second bigger reason is really explainability, right? So we all talk about, we want to do responsible AI, and sort of a corollary to doing responsible AI is really explainable AI. So how can we explain the black box there is, right? So I mean, you could apply a lot of fancy algorithms, the random forest, the k nearest neighbors, et cetera, but I think if we can't explain the features as to why those are associated with the clinically meaningful outcomes, I think we have failed as a community. So that explainable AI is becoming more and more important. I'll show you one example with the pancreatic cancer use case today. Potential to worsen inequities. I think we talked about that quite a bit in a number of talks today about bias, right? So if there's already inherent bias in the data that you're using in your training sets, then obviously that inequity is going to propagate. So how can we kind of think about reducing that bias by either tapping into other data sets or even encouraging data collection, right? So, you know, what I've noticed with some of the machine learning teams is that, oh, we need to validate this with an external data set. Now, journals are asking you to validate with external data sets, but that external data set is not available. So how can you, as a leader in the AI machine learning space, really drive that data collection? So for those of us working in the biopharma industry, the early stage clinical trials are an opportunity, right? So phase 1 trials and phase 2 trials where you have a little bit more opportunity to collect deeper data. Let's design those experiments to collect those data sets so that we could validate what we're finding in some of these larger external databases. We talked about the bias. So I just wanted to put up this slide. I had this from 2019, your left-hand side, from 2019, and you know, already there are over 30 devices, digital devices that are approved by the FDA. But if you look at most of them, they are, you know, either imaging-based or, you know, this whole thing about patterns, right? So, you know, dermatology, et cetera. But of course, in the last couple of years, there are a few that have come out for emergency medicine as well. But we see very few in places like oncology. There are 3 or 4 of them that are approved, partly because of the complexity of the disease, and you know, cancer is not one disease, breast cancer is not one disease, pancreatic cancer is not one disease. So the complexity of the disease and also the types of data that are involved don't immediately lend themselves for solving through a device that uses AI or ML. So we just said that, so it's mostly endocrinology. So in the next 10 minutes or so, I want to give you a very specific use case and how we could potentially leverage AI and ML to really help patients with pancreatic cancers. So this was a patient that we saw at the Georgetown University Lombardi Cancer Center, my previous employer, and he was a 52-year-old African-American male. He lived in the Boston suburbs. He was diagnosed with stage 4 pancreatic cancer. The NCCN, which is the guidelines community, right? So this is the National Cancer Comprehensive Network. It makes guidelines for how we should be treating cancer patients. You know, recommends that for pancreatic cancer patients, you should always consider clinical trials, right? Because the treatments are not great, and you know, the progression-free survival, which is the end point that we measure for whether or not the treatment is working, is less than 12 months for pancreatic cancer patients. So they say that you should consider clinical trials, but which clinical trials? So if you talk to our pancreatic cancer oncologists, they'll say, well, there are hundreds of trials that are available. How do we kind of choose and select? So that was the question we were trying to address, is can we match the patient's clinical phenotypes and molecular phenotypes to eligibility criteria for clinical trials so that we could really speed up the process of identification of clinical trials for patients with pancreatic cancers. So a multi-gene panel was performed on this patient, which helped identify a ROS1 gene fusion. This is one of the molecular anomalies, which is typically not found in pancreatic cancer patients. You'll see things like KRAS and P53, but thank you for telling me it's cancer. You're always looking for these unique molecular markers, and so once the ROS1 gene fusion was found, it was a very quick match to the Star Trek trial. And this patient actually sent an email to our patient coordinator saying he's working again and he's playing squash. He was actually on his way to hospice, but this multi-gene panel testing and this matching to clinical trials saved him from going into hospice. Of course, he passed away 7 months later, but his quality of life was very much improved. So the question is, how do we scale this to other patients with pancreatic cancer? Every patient that has pancreatic cancer should have this opportunity globally, not just in the United States. And if we look at the types of data that we need to incorporate into these models, I have listed a few. I won't read all of them, but it's not just the clinical data and the molecular data. There are also guidelines. There are approved regimens, and there are always combination treatments for oncological treatments, and more trials, clinical trials are emerging. And very simple things like, you know, we tend to kind of look at these complicated features when we build these models as the molecular and the clinical. The thing that was most useful in this database, and I'll show you, was actually how far is the clinical trial from the patient's home, right? And these are extremely sick patients. So if you actually think about, you know, for them to have to travel like 1,000 miles to get on a clinical trial, that's not even an option. So a very simple feature that went into the model that predicted the trial that they actually enrolled to was actually the distance of the center that had the clinical trial from the patient's home. So again, this kind of brings us back to that whole explainability aspect, and you know, how do we really bring these complicated technologies like AI and ML closer and closer to patients? So very quickly, this is the workflow. I'm not going to go into the details of the model, but very quickly here, the process was, you know, this was in close collaboration with PanCan. This is the Pancreatic Cancer Advocacy Network. Now, why did we collaborate with PanCan? So we saw that one patient at the Georgetown Lombardi Cancer Center, PanCan hears from over 50% of cases in the United States that are diagnosed with pancreatic cancers. They're a patient advocacy group, so they have this PALS network. So it immediately scales us to thousands and thousands of patients who are diagnosed with pancreatic cancers. They have a system to obtain consent, so we can go beyond just the clinical data and collect molecular data on these patients. And we use some very simple multi-gene panel tests like Foundation Medicine, and Caris was another company and another test that we used so that we could corroborate the clinical diagnosis alongside the molecular data. And all the data that I mentioned in the previous slide, the guidelines, the clinical trials, the molecular data, was collected in this virtual molecular tumor board knowledge base. So we created this knowledge base which had more than 51,000 rules, right? So essentially, and I'll show you the different types of rules. They were categorized into 3 categories, basically. So I'll talk about the rules in a second for this expert system. But the key, really the key for the success of this program was actually to have the machine spit out the best clinical trials for these patients and to put it in front of clinical experts, right? So we established a clinical team of pancreatic cancer oncologists who understood sort of the molecular parameters and the clinical parameters. There were pathologists and radiologists, and this team was created. We called it the MRP, the Medical Review Panel, and putting this data in front of them and having them kind of upgrade or downgrade the treatments, right? So if the machine recommended a certain type of treatment, so the patient had an mTOR mutation and therefore everolimus was the treatment that should be given to them, you know, the machine recommended everolimus, and then the medical review panel would say, hey, you know, in the last couple of weeks in JAMA, we just saw that there were severe adverse events for everolimus. We're going to downgrade everolimus, and another treatment or another trial was upgraded. So that knowledge then went back into the knowledge base so that the machine was continuously learning, not just from new data, but also from information from the expert clinicians who were reading papers, who were sitting on panels, and they were hearing about why certain therapy was not working. So kind of really that human in the loop and connecting the data to humans rapidly allowed us to put this process in place. So here is the expert system, the 3 vectors for how the ranking was done for the clinical trials. Basically the rationale was the molecular rationale. So we saw the ROS1 fusion. If the patients had ROS1 fusion, then there were certain types of clinical trials. If they had mTOR mutations, they had certain types of clinical trials. So that the expert system kind of created these rules that matched the molecular data to the clinical trials based on the eligibility criteria in the database. Then there was disease relevance. So is this trial specifically for pancreatic cancer, adenocarcinomas, or is this a basket trial that was just testing for mutation across multiple diseases? And you know, we can talk about, you know, whether or not somebody should enroll into a basket trial. These typically tended to be earlier stage studies, phase 1 or phase 2. Made sense for some of these advanced pancreatic cancer patients to enroll onto a phase 3 trial. So that disease relevance became very important. And the third was patient history. Have they ever taken this class of drugs, right? So if they have taken this class of therapy and if they had an adverse event, then maybe, this is not the right treatment for them. If they didn't have an adverse event and they tolerated that class of treatment, but they still did not respond to it, then we need to think about a better class of treatment. So basically it was these three vectors: the molecular rationale, the disease rationale, and then the patient history. So what we found was that this system saved a lot of time when we had the clinicians who were not supported by this virtual molecular tumor board, and they went off on their own. They were looking at the molecular data, the clinical data, and then they were trying to find the right clinical trial for their patients versus having the machine kind of support them. And what we found was that on average, it saved about 45 minutes per patient per clinician using this model. Oh. Okay. So this is just a quick picture of the inference engine, and this is what the rules engine actually captured, and this is all published in Pishwain et al. In JAMIA Open in 2019. So if you're interested in pancreatic cancer, interested in types of data that was incorporated into the virtual tumor board, I invite you to read that paper. So let me talk about sort of how do you democratize access to these complicated systems and algorithms to end users, right? So there were two types of reports that were produced through this complex process. One was the physician report. So for those of you who are familiar with sort of the molecular reports that get generated on cancer patients, they're about 60 pages long, 60, right? And the physician has less than 6 minutes to review them before the patient shows up in their office, right? So what we learned through this process was it was extremely important to have a very good summary page, right? So this was it. They were only looking at the front page, and we had to pack that with the most relevant information. So, teach them a little bit about the molecular pathways if there were residents or, young clinicians who wanted to learn more about how to use molecular medicine. Have something in there that sort of grabbed their attention and teaches them. So that was that pathway diagram on the right-hand side. Show very clearly what the genomic findings and the molecular findings were. We actually did an eye tracking analysis. So using an eye tracker, you kind of can see where the physicians were actually looking at the report, and they went straight to that table at the bottom. They went straight to which are the trials that I can put the patients on, or what is the off-label treatment. So understanding how our consumers use this information can help us sort of highlight the explainability and improve the explainability of the data that comes out of these models. I won't explain the colors, but basically what's off-label therapy, what's clinical trial, and how far these clinical trials were, so that in the joint patient-physician decision-making, they could actually have a conversation that was meaningful for the patient. What was also important was to create a 6th grade reading level patient report, right? So this is exactly the same report that the physician saw, but it was using layman's terms and very simple, sort of a 2-page report. Because if you talk to these patients with pancreatic cancers, the immediate thing that's on their mind is, my disease is going to progress after this disease, after this treatment. So what is my next trial that I'm going to get onto, right? So that's what they're looking at, and it actually told them, this is your next one. After that, this is your next one. It gave them 3 or 4 trials, and then it also told them where these trials were, et cetera, which they understood very clearly. Protera was the company that we collaborated with that did the molecular analysis and the data integration with the clinical data. So this was the scale up. So in a matter of about 3 years, we went from that one patient in the Boston suburbs to almost 1,500 patients across 300 different hospitals. And this was because of, well, not just the virtual tumor board, but also having the medical team that was able to quickly select and put patients on clinical trials. Now, obviously this is just in the United States, and we want to make this more globally accessible. A lot of the methodologies are described in the citation that I've just listed. Won't have time to go into the details of the molecular analysis, but the bottom line was patients who actually received this Protera report, right? So this physician report they lived twice longer than the ones that did not have this integrated report. That's what this graph here is showing. But of course, it's an observational study, and it has to be tested in randomized control trials. The graph in the middle is just showing you the various molecular anomalies that were matched to the different trial patients. So when the story was all told, it was 1,725 patients, so 1,725 patients that were touched by this report, and they took advantage of it for the right clinical trials to enroll onto. Just a quick statistic, only about 3 to 5% of patients with cancer actually enroll onto clinical trials. So systems like this enable them to get onto clinical trials more and more. In fact, after COVID, that number has even dropped in certain cases. So systems like this can enable more patients to get on clinical trials. That's the only way we can make better therapies for these types of difficult to treat cancers like pancreatic cancer adenocarcinoma. So finally, just wanted to quickly thank all of our collaborators at Georgetown and the 14 other centers that were part of the PanCan network that recruited patients, and then Prathera and the medical review panel that collected all the molecular data and the clinical data, and were also the clinicians who actually provided feedback on whether or not what the model produced was accurate and if they're using that information. This was funded using the PanCan's Research Acceleration network, and also by the National Library of Medicine. Thank you very much for your attention, and I am at time and happy to take any questions. Thank you.